everyone. My name is Joy Afolabi, as she rightly said. I'm a graduate student in Dr. Ashbrook's lab, who we already know, and I'll be talking on machine learning, behavior, aging, and Alzheimer's disease. So I'm in the preliminary stage of my research, so I'll just be talking about where I am and where I'm going. Okay.
And so aging is something that happens to all of us, and as much as sometimes we wish we can pause the aging process while still alive, there's nothing we can do about aging. And as aging sets in, usually old age diseases like heart disease and Alzheimer's disease also set in. And what we can do is to try to understand the aging process and how to, mitigate these old age diseases so as to prepare treatments. Alzheimer's disease is a disease we are already familiar with, and it's a disease that destroys brain cells. And we're not sure what exactly causes this disease, but it's been proposed that buildup of neurofibrillary tangles and amyloid and peptide cause the disease. And aging is the major risk factor for Alzheimer's disease. And as more people age, more people are getting older these days, so they have more proportion of people coming down with Alzheimer's disease. So it's good if we can find treatment as soon as possible. AD is a complex disease. Of course, this is a complex disease. So AD is a complex disease, and over 70 genetic regions have been linked to Alzheimer's disease. We're not sure of the roles that these genes play in the disease progression. So that's why we need model systems to be able to understand the effects of these genes. So Alzheimer's disease not only affects memory, but it also affects language, reasoning, and social behavior, and that's where my research comes in. The different genes we believe might be altering the different aspects of these behaviors. So that's why it's important, why we need to quantify these behaviors. And we'll be using mice in my research. So usually, traditionally, the assays that have been used have been collected manually and analyzed manually also, but we are looking to use computers, basically computerized behavior quantification, to be able to explain and the genetic aspects of AD, and hopefully to find new phenotypes that are associated with Alzheimer's disease and aging. So to do this, I'm going to be using machine learning, which we are most likely already familiar with. We've heard of ChatGPT and other technologies. So machine learning is a subset of artificial intelligence, and machine learning has a subset called deep learning. And deep learning has other subsets: supervised, semi-supervised, and unsupervised learning. I'll be using supervised learning and unsupervised learning. So in supervised learning, I basically train the computer or teach the computer what to learn, the software what to learn, and then this software is able to learn from what I teach it, and then it's going to be able to perform the task I want it to. But in unsupervised learning, just give the software the data we have, and the software finds patterns in the data, and then I have to go back and make inferences from the data. So I'm going to be using both supervised and unsupervised learning. Then this is the neural network, and it's basically what makes the computer able to learn from the patterns. So we have the input layer where we give the data into the software, and the hidden layer that helps the computer learn, and then the output which we're interested in. So the way computers are able to recognize images is through assigning numbers to colors. So computers see images as numbers, and from the images the computers see, they're able to recognize poses, and from poses we're able to get pose estimation. And pose estimation is just basically tells us what labeled body parts are where, and then behavior can be extracted from pose estimation, and we will then perform QTL analysis to find the link between these behaviors and the genes. So I'll be focusing on gait and frailty. So different behaviors have been associated with Alzheimer's disease and aging, but in my research, I'll be focusing on gait and frailty. And we talked about how this behavior assays were collected manually. So this is like an example of a setup, and if we were to do it manually, would have had to just sit and observe the mice, or the mouse, how it moves, and we'll term the correct alternation as it's moving from one arm to the next. And an incorrect alternation will be if it keeps going back to the arm it came out from, because you know that mice generally want to explore previously unexplored places. So that would be an indication of cognitive deficit. So that would have been how we would have had to collect this data. But now we can use computers. So that's what we're going to be doing in my research. So I'm going to be using the ABXD mice, which is just basically a cross between the B6 5X pod and the BXD strains. And we're also going to be looking at age, sex, and transgene effects. So we're working with both male and female mice, and we're working with 6-month-old and 40-month-olds, basically middle age and young mice. And then we're also, of course, going to be looking at transgene, non-transgenic, and transgenic mice. So we already have videos of mice in the maze, and we also have the phenotype, the genotype of the mice. So we talked about how the computer will convert these images to values, and then we have software that we use to extract the behaviors, and then the QTL mapping will also be done. So these are the software I'll be using. Deep lab codes, which basically converts the videos into pose information, and single behavior analysis, which we use to convert this position information to behaviors. And we're using R to compare between and see if there are any sex, age, strain or transgene effects on the mice, and then using gene network for QTL mapping. So I'm also going to be comparing, I talked about supervised and unsupervised learning. So deep lab cuts and HRNets are both supervised learning methods, and Mostly enable both unsupervised learning methods. And then HRNet has been used by another group in a different setup. That's the open field setup. So we're going to see if it works in our setup. That's Snowflake Maze out. We'll see that in the next few slides. So I'll be comparing between both to see if maybe there's some traits or some behaviors that one learning type leaves out and the other one finds, or to see which one is more suited for my research. So at base level, this is how the input data looks like. So this is just for one video. So from one video, the software extracts different frames. So the frames are basically like screenshots of different scenes that are important in the video. So each frame has its number, and then each body part has its position, the X and Y position, and its values. So this is basically what the computer learns, and then from what it learns, we can then use what it's generated to work on other videos in the same setup, of course. So this is the setup we're using. We call this Snowflake Maze. And these dots are basically the key points, and this is what helps the computer learn what position and that are important. So this is how it looks, the mice being tracked in the maze. And we can see that the mouse moves from one arm to the other. So we're talking about correct and incorrect alternations. So we can see that it moves, so it shows that it's basically a healthy mouse. But we also see that in between, it rests itself. And traditionally, that wouldn't have been taken into cognition. But with machine learning, we'll be able to look at and extract behaviors from these patterns we see. So this is a preliminary work from the lab, and it's in different setup, but we're already seeing sex effects and transgender effects. So like in the BXD 102, we can see that the males perform better than the females. And also in this BXD 29, we see that the females perform better than males. So we're already seeing sex effects. But in BXD 60, we can see transgenic effects. You can see that the non-transgenic mice perform better than the transgenic mice. So just to tell me that I'm taking a step in the right direction. So yeah, in summary, we're trying to identify new phenotypes that we might be able to link to genetic regions of the ADBXD mice. And we're also trying to basically do what has been done manually with computers. And we're hoping that we'll be able to contribute to the understanding of AD mechanism. And also, of course, the end is to find new therapies for disease. I'd like to thank my supportive mentor, Dr. David Ashbrook, and members of the lab, and all members of my committee. Without, I'll take questions.